So I'd like to just kick off the discussion with a question for Terry, because I think one of the absolutely key elements of a 3D cell modelling framework is the ability to do the model reduction process, depending on the question being asked. And how much thought have you given to the process of taking the very detailed synaptic analysis, molecular-based analysis that you've done, to extract from it the key phenotype that you'd want to describe if you were then incorporating that synaptic model into a larger scale?
Yeah, that's a key question, and we've started, I should say, to integrate the Mcell particle-based approach with the neuron model at the next level, which is a cable model, a multi-compartment model that keeps track of all the electrical properties of the synapses and the dendrites. And the way we're going about it is to describe within the neuron model not the actual details of this particle simulation, but rather a summary that just captures in a relatively few parameters the output of all of the detailed simulations, so that we can keep track of, say, 5 parameters that describe the strength of the synapse, the short-term dynamics, time constants, and those are now parameters that are different for different synapses and their histories. So you're right, you have a different model for every single level, and you have to go from the number of degrees of freedom is collapsing as you go up, but you have the richness at the bottom to draw on whenever you need to go back and find out, what's actually going to happen. And with respect to, I actually had a challenge there, which I never actually completed, which is how are you going to deal with 10 to the 21st states? The way that you handle it is by the fact that in a particle-based model, the CAMP kinase 2 can only be in one of those states at any given time. And it can only branch out to, at the next time step, in a relatively small number of possible states that are going to have certain probabilities. And so it turns out that you don't have to have 10 to the 23rd equations. You just need to keep track of where it's going to go next. And so I think there are going to be simplifications going up and going down that will allow us to computationally capture all that complexity.
And is it obvious what the modules are at each level? Because that's
No.
one of the key aspects of building a multi-scale framework, is identifying the appropriate modules for.
Well, there are structures, anatomical structures that give you hints, like the synapse and the spine. But it's clear that once you really explore a particular level, there may be, depending on the question you ask, there will be different modules that you may need to pass up. So it's not a single model that it will answer all the questions. It's a subset for every question.
So I think the key to this problem is to, automatic model reduction is a fantasy right now. You have to take it case by case. It may work in certain applications. Usually it doesn't work, so you have to do this manually. I think the key here, how to extract parameters which could be transferred to a model which operates at a higher scale, is to run virtual experiments, perturbative experiments, and see, what kind of quantities you are interested at the higher scale level, and try to, build thousands of simulations, measure them on a computer, right? Propagate them up, and this way you can build a systematic workflow which can take you from molecules up to the higher modeling scales, and that's the best bet right now.
Yeah, over there. Yeah.
Remember to say your name too, please.
My name is Sophia. My
Press the bottom button for a second or two. Okay.
Perfect. So as computationalists, how do you deal with results that challenge experiments. So how do you validate and sort of not try to fit experimental data, wet lab data? And I mean, you can't ignore it, but at the same time, it could be misinterpreted. So I think that's a really big challenge, and I'd like to see what the panel thinks about that.
Well, I think that, that's a very good question. I'm not going to talk too much about validation, but one interesting thing that simulations can do is they can simulate experimental hypotheses. So for example, you run your simulation, you discover something unexpected that experimentalists have not seen. Now the question is it real? Can we test it in the lab? So I think that, of course, experiments, simulations, it's nice if they are predictive, but they also can serve as a hypothesis generation tool. So I don't know, anybody else? Any comments?
Yeah, I mean, I guess for me, definitely, I don't remember who it was yesterday, but someone was talking about how it would be hard to get experimentalists to be interested kind of in the simulations that we did. And that was an interesting experience for me when I was doing my PhD thesis. I had this very model that I thought was very exciting. And I showed it to a well-known experimentalist who I thought was going to just jump out of his chair and run to the bench and try to validate it. And I realized that of course, wasn't going to be the case. But probably the best thing that came out of that is I decided to do the experiment myself. I think that since that time, I definitely think that there's this real interplay, like I was trying to talk about here just briefly, but I think there's an interplay between the simulations and the experiments that can really be exciting, you know, if they're occurring with kind of, in my case, it's in the same hands, but it certainly doesn't have to be. But I think that as you bring those things together, right now, for example, what we're trying to figure out, so the model, I showed some of the kind of most interesting things we've seen with resolving the model failures, but there's actually a whole class. We've divided maybe 4 different categories of model-driven discovery. One is just the model's totally wrong, right? One is go back and look at the experiments. A really interesting thing that we saw in our model that I didn't show. So we can't get the, well, let me put it this way. So 50 years ago, they measured the chemical composition of a mycoplasma cell, and they weighed, essentially, the percentage of DNA that's in the cell. We don't actually need that data point anymore because we have the DNA sequence, so we can just add up molecular weights. And it turns out that if you take those 2 measurements, they're off by a factor of 5. So the chemical composition that was measured back then is not enough to make a 1/5 of the chromosome that we get from the DNA. And that's just a very simple example of how, you know, once you try, and then there was the other comment someone said yesterday, which is that you don't even have to do a simulation with a model. So we kind of recognized that just as we were building it. Before we'd done a single simulation, we realized, wow, these data sets are totally inconsistent, you know, and there are many, many cases of this. So I guess it's kind of a rambling answer, but there's definitely some very interesting research to be done in trying to really deeply mesh the models of the experiment.
So just as a footnote to that, I mean, it's clear that, you know, if you really believe in it, do the experiment yourself, but not everybody has a lab.
Right.
So that's a real advantage. But the next best thing is to have a very close collaboration. And I gave you an example with Mary Kennedy and CAM kinase 2 and that whole pathway. And it's been very, very fruitful over the last 10 years, both for our lab, getting the numbers we needed, the biochemical reaction rates, but also for her. So Mary Kennedy now is running Mcell herself as an adjunct to doing experiments. She actually helps her do experiments, and she's learning Python. So it's possible, I think, to close the cycle if you have a really good working relationship. Yeah. And ultimately, and I really like your story about the reaction that someone had about, you don't expect me to do the experiment, do you?
Yeah.
Is that the moment at which they're going to want to do the experiment is when they see their competitors having an unfair advantage by using the model. That's when you're going to turn people's heads. And they're going to say, wow, we need to bring those people into our lab so that we can have that advantage.
Yeah.
We're not quite there yet.
Yeah.
So
Phil,
I just
Phil,
wanted to
yeah.
quickly comment,
Sorry.
comment on that, sort of doing both the computational and the experimental part in my own lab, I kind of see that challenge. Because, you have to really teach the students, to let the data speak, and they have to really, objectively go in, look at the data, and then formulate their hypothesis, based on what the data is telling them, and not go in with sort of preconceived notion as to what is important. So, and sort of getting that across is very critical. So, yeah. So I think drawing on my own sort of experience, I think that's very critical.
Phil.
So I actually just wanted to build on this. In fact, you answered most of the kind of things I was going to ask, I think. But I would just cast the question as one between experiment and theory and the people who specialize in those respective areas as getting to a point of building trust, and building greater trust. And collaboration seems to me a very vital way to build trust in the way that you've described. And I think for the older ones of us, we recognize this tension that existed many years ago in the early days of bioinformatics and so on. I think most of that's, thank goodness, gone now. And this is, the new wave, I suppose. And it's a question of how we can short-circuit that tension as much as possible. And the bit you didn't mention explicitly is where this kind of resource might play a role is in education. And obviously you're training people now that, when that generation kicks in, a lot of this will go away. But maybe there's something here, and I'm just interested in what the panel can comment on that what we might do in an education way to sort of accelerate the process. And that could be courses, it could be online materials.
Yeah, that's excellent question, because clearly we need education here. I think there's lots of simulation tools out there. How many of those tools are easy to use? How many people could reuse them in their classes? There is not that many. So I think there has to be probably community initiative to develop a course curriculum on multi-scale modeling of going from molecules to cells using available software, because this is the key. I mean, instead of actually teaching raw equations and asking students, okay, you solve this equation in Mathematica or MATLAB, yeah, let's use the tools that we as a community developed. And I'm sure that all of us here are doing a fair share of that. We're organizing Mcell workshops. Peter is having his workshops. So, but it's not enough. I think what's missing is just like I can pick up a MATLAB manual, which comes with software. So there are examples where I can work out. We need something like this for this community, for cell-based modeling, for single-cell modeling, molecular dynamics, and so on.
So there's another pathway. And has anybody here played SimCity? Why don't we have Mcell? I mean, you have these beautiful ways of visualizing, David, and in your work. Why not create a game in which, you're exploring inside of a cell, and that you have to have a story, but the point is you can explore the nucleus, and here's the DNA, and you can see things happening, and it can be made into a mystery. What's wrong with this cell? I don't know. I mean, that's going to capture the imagination of a whole generation, I think, if you could get them the information surreptitiously through that pathway.
I'm working on a couple different game projects, actually, and using autofill, autopack as an engine for the underlying system to develop scenes and worlds that people can explore quickly and stochastically, so they're novel every time. I also want to suggest that if we are taking these types of experimental procedures out of the individual labs and putting them into a bigger context of this virtual cell project, that providing accessibility portals at every level to the different types of simulations that are available, and thus exposing transparency of what your confidences are, and letting sort of effectively crowdsource your an iterative improvement from the community as a way of building trust from the community, I would suggest.
So I have a question up here. Jan Powell, College of Nanoscale Science and Engineering. I'm curious for the experimental relationships, collaborations that you have, what do you look for in a collaboration, and how often do you expand from the initial collaboration to establish new collaborations while keeping some trust and sincerity, effort relationships with the first collaborator?
So let me ask a question. Are you married?
No.
Okay. Well, that's what it's like. In other words, it requires, if you're going to be working together for many years, and it takes at least 5 years before you're actually on the same page in terms of understanding each other. But you have to like that person in order to be able to go that route.
Yeah, Eugene. Yeah.
So, definitely, some of us doing something wrong.
It goes off.
No. Okay. So I'd like actually all of you guys on panel, including you, Peter, if that's okay, answer this simple question. So say we are 5 years from now. What do you think is doable as a community effort with regard to 3D virtual cell? For example, this is just example. Can we do beta cell? Marcus, can you start?
Sure. So I guess it's hard for me to answer the question directly because I'm not as, I mean, I did molecular dynamics as an undergrad, but I haven't gone back. I was very impressed by many of the things I've seen here, but it's hard for me to take that into context how long it took, right? So I looked, for example, at Terry's thing today, and I thought, wow, that looks like that would take forever. So, those things look very challenging to me. Using our approaches, I can maybe say some things about that. So using our approaches, we're going to go to E. coli, okay? Then we're probably going to go to some kind of a eukaryote. We do a lot of work with macrophages in the other half of the lab, so I really like the idea of going after a macrophage model. So we're planning to do those things, and I see those as I see us having, let's say, a decent E. coli in 2 years and maybe a decent eukaryote in 5. I don't think that's unreasonable. For those things, we are going to add more space, incidentally. So we've worked out a framework to include detailed PDEs in these models, but it's not molecular. I was really impressed, saw Bernard, my old PI, he has been adding the structures with Phil, and that's been very interesting. So we're thinking about all those things in about that time scale. Yeah.
So yeah, I think that it's really great to be ambitious, right? Because if you're not going to get anywhere. But if you're too ambitious, that is to say, if you aren't realistic about where you're going to be, then you may not be heading in the right direction. And one of the things that I've discovered in my own career is that life is what happens to you when you're planning to do something else. That is to say, you often can't predict what you're going to be doing 5 years from now. Mm-hmm. And part of it is the fact that new opportunities occur that you have never have anticipated. And one of the driving forces behind all of the research that you saw today was the fact that computers have been increasing in power exponentially. And it's, by the way, it's behind almost everything that's happening today in terms of big data and data collection. It could never have been done without computers and computer storage and the internet. And so really what we should be asking, where is the computer system? Where will computers be in terms of availability to us as a community? Not now, right now, but say 10 years up the road. And what could be done with that? Prepare ourselves for that day when it comes, because it'll be here before we know it. And so, we try to do what we can with what, the resources we have right now. But you really do need to plan ahead. Like the simulations I showed you with Mcell really couldn't have been done 10 years ago in a reasonable amount of time. But now Tom Bartol does these simulations. It only takes a few minutes, and 10 years from now, it'll only take a few milliseconds.
So taking that whole into account, without being right on the spot, just guesstimate. In 5 years, what do you think is doable?
Well, I agree that we should be in the position to be able to look at the complexity of a whole cell, 3D. There'll be hybrid models. There'll be different. It'll be question-driven in terms of specific disease states and so forth. And I actually don't see any barrier. I don't see any showstopper. It's just a matter, again, of time and effort and resources. And I think the community here, I'm really impressed with how much community we already have. I mean, it's not a matter of building the community. It's there. It's integrating the community, getting a lot of the tools that we have developed to be working together and useful. We know there's a lot of advantage to doing things in multiple ways because every method and technique has limitations. And so if you can come at the problem from many different directions, then the intersection is going to give you something that's much more powerful than any single tool.
Maybe one quick thought, I promise it'll be short, but along these lines, so when coming up with a proposal or project, like it's important to know what to promise. Like I think I would greatly prefer that we be able to deliver and even deliver more, as opposed to, we mentioned, for example, the Alliance. It wasn't necessarily their fault, but I mean, it's a tougher situation when it doesn't pan out the way you think, or for example, several years ago, of course, the promises that were made surrounding stem cells. It's going to be, it can definitely have a backlash, and it's, again, about creating the trust. I think it really is important. I'm a big fan of over-delivering, and so just a thought to throw out.
So I think that what's doable within 5 years is this collection of components, software components for biological modeling. And I mean here, SBML model solvers, PDE solvers, which are not maintained by hundreds of institutions, but they are in one place, and they come with one-click installer. And it's simple task, which can be delivered within 5 years. I mean, I'm sure that some of you can do it in 1 month, but at the very least, if I can have all the components, I could start building my computational models in much easier way than it's now.
So historically, there was a study that's been done. If you ask someone how much can you do in 5 years, they always overestimate by a lot because you're ambitious. But if you ask them how much you can accomplish in 25 years, they always underestimate. And the reason is that's the time scale for a major discovery or new technique to occur that allows you to do something you couldn't even imagine before. The whole genome, right? That's something that wasn't, in fact, I once asked Francis Crick, did it ever occur to you when you discovered DNA back in the 1950s that within your lifetime that we would know the whole sequence of the human genome? And he said it never even occurred to us to ask that question because it was science fiction, right? So, I mean, in your own lifetime, things will happen that are science fiction. And you can't predict. That's the problem.
That's a great point.
So we're doing we're doing an experiment right now with releasing this HIV model, which is our baseline experiment. It's a simple system, but it has all the components of fibrous elements, transmembrane elements. And we've got a 3-month timeline for this online contest, which is, again, participants between scientists, artists, engineers, et cetera, contributing to this project. If that works, so I'd be able to answer in 3 times, it should scale to, we've got all the basic building blocks to build molecular model of a cell. And then the question is, for the bigger group, how is that useful to them? AutoPack, AutoCell can pack anything into anything. So if you're doing incredibly coarse-grained modeling, we can parameterize the cell for you and position the objects. You don't have to do use 50% of your computational time just to relax the system for Brownian dynamics, for example. That can be done in a matter of seconds. And all those components can be set up. So one of the most critical things that I would love to see at the end of 5 years, actually at the end of 1 year, is a framework that allows us to tie these packages together, a file format that allows us to integrate all these packages together, and to build from there.
Okay, so I think I'm the only one left. So I'm going to very quickly answer that, or at least attempt to give my suggestions on that. So So obviously, it's we have a time frame for this. Phil, I'm thinking you're thinking about the next 5 years. What are the deliverables? So sort of one thought would be to kind of focus on the core components of the cellular machinery and sort of try to model that and then how do we come up with those core components? And that's what I think it's important to see what nature is telling us and to kind of look at what evolution is telling us about what is absolutely critical in terms of cellular functions. And then once you decide what the core components are and then focus your first efforts on really figuring out how these core components work together, and then we can then build in finer details into that and then sort of build the model that way. So that's sort of my thought.
Okay, so I think it's reasonable within 5 years to have substantially built a 3D cell framework, anatomically based cell models for a few cell types. I mean, there's about 300 to 400 cell types, but I think it's worth focusing on a few where the questions you could ask really matter. So, for example, in a cardiac myocyte, there's already been a lot of work with electron tomography and super resolution confocal and so on. And I think there are some questions particularly relating to heart failure where the changes in the anatomy of the cell are critical to the phenotype that we want to understand for heart failure. So understanding spatial relationships, diffusion distances, distribution of particular transporters and so on, and as those change under disease conditions is really important for answering very specific questions to do with, for example, heart failure. I think the same could be said for, in a hepatocyte, the understanding of the detailed cytoskeletal structure in an hepatocyte where under, for example, non-alcoholic fatty liver disease, you get deposition of fat that leads to changes in the morphology of the cell, which lead to effects on bile drainage. Those sort of questions, I think, would you could justify putting a lot of effort into really understanding the 3D anatomy of the cell. But I think it's far better to be doing this in great detail with very focused questions that have value in terms of the phenotype that you'd reveal over the next 5 years before you sort of expanded it into a much bigger program. Yeah.
So I would, I think this is a great discussion and a great foundation for talking this afternoon. But and I think the grand challenge, you need a really grand challenge kind of goal from this, and what we can deliver is really important. We're going to educate everybody how to integrate communities and integrate tools. So this will hopefully be something that will teach the next generation how to do this, and we'll educate young people to think across boundaries like that. So I think we have a real opportunity here. And I guess if we look at, I was at the Gilman first meetings as well, and structured genomics as well. It began gathering data, but it left out the biology. One began with the biology and just getting data and not doing the modeling. And so I think we really, from the beginning, whatever we do, we have to keep the biology and the modeling integrated and build those collaborations. I agree with you. This is just, you're not going to have everything in your own. You have to work closely with experts in these different areas. And so that's really essential. And how we can train. Burroughs Wellcome had a wonderful training program for training people at the interface between theory and biology. And how do we get funding up to really build not just the big conglomerate, but individual collaborative projects where, like, you and Mary work together, or this is just a wonderful way you gain respect for each other, and it's a dialogue back and forth where everybody learns. So I guess just your comments, and I think this is a wonderful opportunity that we have.
Okay, I think we should stop there. We're right on time. And can I just join with me in thanking all the speakers for this morning's session?